From Microbe TV, this is TWIP, This Week in Parasitism, episode number 200, recorded on November 2nd, 2021. I'm Vincent Racaniello, and joining me today at the Incubator in New York City, Dixon Despommier.
Hello there, Vincent, and everybody else. I can't tell you what the weather's like because there are no windows at the Incubator.
Well, there are, but they're covered up.
Well,
right?
I can't really tell, but it's a gray day out there. I can just tell you it's a gray day, a little bit raw, no precipitation, but I think the humidity is around 55 to 60%. It does feel that way. And it's an appropriate day for TWIV 200 because it's election day. And
TWIP, not TWIV.
TWIV. I meant TWIV. A TWIP. I meant TWIP. Who's confused? It's election day in the United States of America.
It is election day. Also joining us here at the Incubator, Daniel Griffin.
Hello, everyone.
And joining us remotely from Glasgow, Scotland, Christina Naula.
Hi everybody. It's
Hello
good
there,
to see
Christina.
you all.
That's good to see
We
you,
did
too.
invite Christina to come, but it was just too far for her.
That's right. That's right.
Well, I think the travel ban on UK citizen only finishes next week anyway.
Ah,
So
you can't leave the UK or you can't go elsewhere?
no, I think I can't enter the US unless I have family or I am family or I'm a special person, which I am in a way, but not in
You're all of the above to us.
not in the way needed. But I think that is going to finish on the 8th of November.
I see.
But of course, I also don't have a proper exam, a proper vaccine, so that would be another hurdle. But it's good to be here from Glasgow too. So
Indeed.
Anyway, special episode 200. We thought we'd gather here. We haven't been, Dixon has not been with us for some time, right?
That's true. I've been remote from Fort Lee, New Jersey.
And I've seen, last week, Daniel, that was the first time we recorded together, right?
We're seeing a lot of each other.
Yes, we are.
Yeah. we're almost done with that, right?
I think this is the...
For me, it's nice. It's nice to see everyone again.
Well,
so.
I think we have no plans, but you could always pop in anytime, Daniel. I'm here recording most of the time, and that was part of the feature of the Incubator, that we could be here, right?
Right.
And so I, in fact, got a very long table that we could all sit at.
Yes.
It worked out perfect.
And if no one's here, I have a nice long table to work at.
That's right. That's right.
Which you guys can see because this wide shot is showing that.
Well, we were reminiscing before, Dixon and I, going through when I joined TWIP,
Right.
which was episode 80, and here we are at episode 200.
That's right.
So this is a good time to reminisce. So Dixon and I started TWIP after TWIV. It was the second, I
right.
think. And I said, look, Dixon had started TWIV with me in 2008, and it had been going for a while. And I said, Dixon, your thing is parasites. Why don't we do a This Week in Parasitism?
Yeah.
You remember?
It was a great idea, of course.
And so we started, and for the first 20-some, each episode was a different parasite. Do you
Right.
remember?
Yes, I do.
And then after that, we said, okay, what do we do next? We started doing papers.
Right.
And then at some point, Daniel was showing up.
Now, I said clinical cases are missing in our... I actually said that. And I said,
Clinical
I know
cases,
just
yeah.
the person who would agree to do this. And
But-
he was just down the hall.
He wasn't there from the beginning, right?
No, that's right.
He joined the Golf Lab in
That's right.
Columbia and
That's
so
correct.
forth. And you and Daniel used to talk all the time. He would come in and stop at your desk and chat with you. And then one day you said to me, I think we should have Daniel on TWIP. And I said, okay.
Yeah, so that actually happened after Daniel and I co-attended the annual meeting for the American Society for Tropical Medicine and Hygiene, and I drove down, and he drove with me, and that's when we really got to know each other because that's a 4-hour drive from New Jersey to Washington.
It seems
DC.
longer.
It seems longer. It was that's now he tells me. I get this data all too late. I could have dropped you off in Baltimore. That really would have been shorter.
And so here we are now, 200. We have had Daniel, and then not so long ago, Christina joined us.
Yep.
To give a different perspective from across the waters.
Indeed.
And so here we are, the TWIP Quadfecta.
There you go.
And we had actually planned to have a little audience, but I don't think there's much room in here for an audience. So
No.
I don't think that's in the plans, unless we knock down this wall one day. And make the space
No,
bigger.
no,
But
I
the
think-
space on the other side's nice, right?
Yeah.
To keep
yeah.
separate. So
And actually, there are several athletic arenas not too far away from here that you could avail yourself of if there are no games. Madison Square Garden is about 2 blocks away.
Yeah.
So you could sell seats and we'd have a remote audience.
Madison Square Garden is 2 blocks away. Yeah. You know, one night I was walking to the train, and above the garden, there was a big billboard advertising the next act, right?
Right.
And up there was Joe Rogan.
Really?
Yes. Joe Rogan. And I mean,
Go figure. Go figure.
go figure. And, you know, it's funny. When I was in Austin recording the Lex Friedman podcast, he got a phone call during the podcast. He said, I have to take this. And it was Joe Rogan.
Right. Right.
And he said, "Oh, talk to this guy Racianello. Do you know him?" And I heard the voice. No, I don't know who he is. Okay, I'm waiting for the call.
But he will.
I don't know. Maybe he thought that the Lex Friedman was enough. Anyway, this is TWIP, and so let's do our thing here. We have a case from last time. Daniel, what did we have?
Sure. Let me remind everyone who's tuning back in and tell a short story to those who are tuning in for the first time. The last TWIP 199, we finished off with the mystery case of a gentleman in his 40s who has been having repeated intestinal issues. We mentioned he'd been diagnosed with Giardia, treated, it got better, got it then again about a year later, again not feeling well, comes to see me, actually referred to me by a gastroenterologist because his stool testing had shown blastocystis hominis and Endolimax nana. I was asked to weigh in on what might be going on. So we got a little bit more background. He lives in the New York tri-state area. He is single. He's active socially with different partners. He did not have any medical problems prior to this or other than this. I mentioned he was taking PrEP, pre-exposure prophylaxis, to protect him against getting HIV. He is HIV negative. He often goes into the city itself for these social experiences with the different partners. His exam and labs were really unremarkable. The only remarkable issue were the stools. I mentioned last time that we had gone ahead and we had done a therapeutic trial with metronidazole, which had no impact on symptoms. All right, I think we left it there.
We did.
Christina, can you take that first email?
Yes, I can. Ingrid writes, Dear TWIP team, I really appreciated how Christina's case study for case 199 had the kind of difficulties of diagnosis, red herrings, testing problems, etc., that are probably common in real life clinical practice. That was great and had the side benefit that only a modicum of brave souls sent in guesses, and they all included interesting thought processes behind their guesses, not just pat scrapings of a Google search. I love TWIP, including the write-ins, but sometimes get a teeny bit tired of listening if there are huge numbers of them every time, especially if most of them are the same. Fewer guesses left enough time for a paper. I had caught the news earlier about this first ever successful malaria vaccine and was hoping to hear the TWIP team's reaction. Thanks for covering it. Thanks for all the fun cases, information and friendly discussions on TWIP, TWIF and TWIVO et al. Ingrid from Berkeley. And she carries on to say, big rain all day today. First
Great.
big rain that I can remember in 2 years. Like the old days standard and frequent fall winter rain that I remember from the 60s into the 80s. I'm guessing this is the official end of this year's fire season and crossing fingers for enough more rain this winter spring to ease our drought. P.S. Vincent, I really like the new 2-color background on the wall behind you in the incubator. And I'm just looking at it. Big improvement on the plain white, and the colors are subtle, not overpowering, just right. I actually can only see one color.
I'm sorry, I moved since the last time we recorded.
Right.
I was recording over there. And now there's just a chip. So Christina, behind this picture, there's a chip on the wall, and everybody kept noticing it and saying they thought it was blemish on their screen, so I had to put up a picture to cover it. Isn't that crazy?
That's funny.
Well, this background will change. We're working on it, but we're not there yet.
Right.
Dixon, can
Yes.
you take the next one?
I certainly can. Andrew writes, Kia ora from Pongaroa. Book 1, yay! But I am still case guessing. I am addicted now and looking forward to seeing who wins a book on TWIP 200. I have an idea for the book signing event. Make a film and call it The Signing. You can work out among yourselves who plays the Jack Nicholson part and who has to explain to Amy why there is an axe damage to the door incubator, or to the incubator door. On with the case. From the gentleman who gets repeated cases of intestinal issues, my thinking is that he has what was erroneously called gay bowel syndrome. That's in quotes. It is basically caused by a larger than normal dose of infectious matter that is passed during sexual activity. The dose is large enough to overrun the immune defenses so that an acute infection that normally would be quickly dealt with becomes symptomatic until the body has a chance to ramp up and clear out the pathogen. The sort of activity required is not restricted to gay sex, and I imagine that practicing safe sex would prevent future problems. Nah, Andrew.
Well, just to let everyone know, since we are gathered here today, Daniel and Dixon autographed the 100 copies of Parasitic Diseases 7. And so they are now will be shipped out. I no longer have an excuse, gentlemen, not to ship them out.
Not only that, you're not very far away from one of the largest post offices in the United States.
No, that's no longer there. That's a train station now.
Oh, I take
In
it
fact,
all
a very nice one. Have you visited?
Take it all back.
Daniel Moynihan Station.
I know of it.
beautiful.
That's so silly.
It
of me
is
to
really
not
nice.
remember
Yeah.
that.
No
It's
more
really
post
a big
office.
upgrade.
No,
It's great.
I take it back.
Anyway, so you will be getting your books perhaps by the end of the year.
In due time.
This is
as
like
we
the
say.
Novavax vaccine. There's a very wide confidence interval here on the delivery.
by the end of the year.
That's right.
All right.
But they filed for approval in the UK. Sorry,
right.
that's not
Yeah.
TWIP.
They
but
got
yeah.
approval in
That's
India
okay.
yesterday, I believe. So we're moving
Perfect.
forward. So the books,
Getting
maybe
there.
those books will be coming
Well,
out.
you never know. You never know.
So, Christina, on a recent TWIV, we discussed the Novavax results were submitted as a preprint, and I mentioned that you had been part of the trial.
Yeah, I heard that.
And
It's lovely to hear my name.
Then after I said, maybe that's what do they call confidential medical information? What
I
is
think you shared it actually on TWIP.
Yes.
So,
so that makes it okay.
Yeah.
yeah, you're not violating
Yeah.
HIPAA. Do you have HIPAA rights in Scotland? Do we have to grant you HIPAA rights?
I'm not actually sure what they are, so maybe we do, maybe we don't.
No, nobody is. Nobody is, Christina.
Right, okay.
All right, Daniel, why don't you take that third and last one?
Certainly. Martha writes, Dear TWIP, I will be brief. I just got the heads up at the online virology course that a new TWIP was coming up. Of course, I am still sending guesses, although I want to book a few episodes back. The case of the man with the complaint of recurrent abdominal discomfort, no other complaints, treated for Giardia in the past, treated now with metronidazole with no resolution. His exam was otherwise normal, labs only significant for blastocystis and endolimax nana. At some point, it was held to be true that blastocystis was not a pathogen and did not cause symptoms in immunocompetent persons. But it seems some immunocompetent persons do have symptoms. It also appears that there are some blastocystis that are resistant to metronidazole, so perhaps a course of the alternative treatment, paromomycin. Wishing you all the best, Martha.
And that, listeners, is it for our guesses.
3.
3.
I know
One has already won a book, so there's actually just 2.
Wow. Well, I know we had someone saying there were often too many guesses and we weren't able to,
Now, this is a bit of a stumper,
Actually-
I must say.
Dixon, two
Yes.
of these 3 people have won books.
2 out of 3.
Oh my gosh. So we
Well-
don't even, there's even a number generator. You just win.
The point is,
Ingrid wins by default. There you go.
that's
everybody.
right.
Ingrid, send me your address, twip at microbe.tv, and you're in California. That's all I need. And perhaps by the end of the year, you'll get one of these freshly signed
That's right.
volumes.
That's right. So now I guess it's up to us to add our little 2 cents
Yes.
worth. Well, I mean, I was obviously I'm old enough to be part of the scene when the AIDS epidemic began, and I was smack dab in the middle of the infectious disease units because obviously my subject is parasites. So, being the only person at Columbia's Medical Center that knew anything about parasites, other than clinicians who had treated them, I was always included into these grand rounds, and I learned about the development of the gay bowel syndrome and how prejudicial that term is, and then how allergic we were to exploring further what the causes for these people's demise was. And you know about these public displays of dismay the band played on, et cetera, to let everybody know that if you're of a certain social group, you are basically excluded from basic medical care. And that all's changed now, but it took an epidemic to cause that to happen. So thinking back along those lines, I think one of the guesses was spot on. And that was the one I read because, indeed, gay bowel syndrome was later on changed to a condition which means that your body just becomes overwhelmed with all kinds of non-infectious and infectious agents. They don't have to be infectious. They would just be microbial, part of the microbiome of another person. And as a result of that, there is these transient syndromes which don't add up when you look at what they mean. Did you have fever? Did you have diarrhea? Did you have discomfort? Did you sleep? Did you not sleep? All these, and everybody had a different story with regards to that. And so it became very confusing as to what was going on until they finally figured it out. So I think what you're really looking at is an overwhelming load of microbial life through the oral route that induces temporary changes in a person's basic symbiosis with their own microbiome, and it causes disruptions, which results in these constellations of signs and symptoms, undefined, untreatable, and just stop doing that and practice safe sex, and it all goes away.
So, Dixon, you don't think blastocystis and endolimax necessarily have anything to do with this?
Well, with repeated negative results from metronidazole, it's unlikely that both of them are resistant to metronidazole. He's just reacquiring them as he develops his partnerships throughout the year. So, because those are two common non-parasites that inhabit the human gut tract. So it's likely that he was visiting some of the original people that he might have acquired those microbes from to begin with. That's my view.
Okay.
What do you think?
Should we let Christina jump in? Christina, you want to jump in from Glasgow?
I'm on quite thin ice with that one, to be honest. I don't really know much about either blastocystis or endolimax, but looking at the life cycle of blastocystis, I thought it was hugely interesting, and I think maybe be a parasite that we could cover.
Yeah, should we spend a moment just talking about what is the story with blastocystis? Remember, we used to do a good job of saying, all right, we've mentioned it, let's go through the life cycle. So, Christina, shall we let you?
Yeah, I'm just trying to actually make it a bit smaller on my screen so I can see it as a whole. I'm looking at the life cycle actually on the CDC, because I don't think you're showing a life cycle in. No, you don't show a life cycle in parasitic diseases 7. So it's okay to have a look at the CDC one,
yes.
I think.
I think we don't show it. We don't show it because we don't think it's a parasite. That's basically why we don't show it. We didn't show endolimax nana either, right?
Yeah, I can bring that up as well. But actually, the first sentence that it says here in the blastocystis life cycle is that the life cycle of blastocystis is not yet understood, including the infectious stage and whether and which of the various morphologic forms of this polymorphic organism that have been identified in stool or culture constitute distinct biological stages in the intestinal tract of the host. So actually not very much is known about it. And if you have a chance to look at the life cycle on the CDC website, I think all you need to do is put in your Google search CDC parasites A to Z, and then you can follow the bee and find it there. Actually, it's got lots of really interesting life cycle forms. So it's got in the intestine, we have an amoeboid form, and then a form that is called granular. another form that is a mitotic form, which presumably divides by binary fission, and a vacuolar and a cyst form. And these latter two forms are the ones that one could possibly find in feces, and that are thought to be potentially infectious. So, but there's really not much known about it at all. And I don't know if Dixon, with your many years of experience, maybe you can add a bit to that sketchy life cycle.
Yeah, best way to put it,
year.
Christina, best way to put that is many years of inexperience, because it's been a confounding organism for many years, and you've got multiple vacillating opinions about it in the literature. Some say, "Oh, yeah, it's a pathogen because I have a patient that had this and they got very sick, and then I treated them and they got better." And then other people say, "It might be a pathogen, but I haven't been able to treat it because it didn't respond to anything I gave them." It's been cultured, and it's an anaerobe. That's why they used metronidazole, thinking they could knock it out, but that did not. This is probably one of the most resistant anaerobic infections to that drug, because bacteria even respond to metronidazole. And it's used for treating amoebiasis and virtually other anaerobes like Giardia as well. So it's a conundrum, basically. That's what it basically boils down to. And I think there will never be any definitive results from this, because it's very, very difficult to find only blastocystis in a patient. And you can see right now that Endolimus xana accompanied that protozoan. And so, you're stuck with trying to choose between which one was causing all those symptoms, and the answer was neither one. So that's why we didn't really think of including a life cycle. A. B, we didn't know what the life cycle was, so how could we include it?
What about Vincent? Do you have any comments? Did you want to jump in?
Daniel, do you remember TWIP 99?
Uh-oh.
That's what I was probably going to bring up, because we talked about an interesting paper, and we talked about the fact that the genetic diversity of blastocystis is about as broad as the genetic diversity of gram-negative rods. I mean, it is an incredibly diverse group of organisms that we're calling blastocystis. So, and in that paper, I think this was the episode where we discussed the paper, it looks like there is a certain subset of blastocystis organisms that might be the ones that might be, I keep qualifying here, might be pathogenic. And so they did studies where they looked at, this is often the case. You give people therapy, doesn't do anything. But other people, you give them therapy, and coincident with therapy, they get better. And so they were looking at the genetics of blastocystis, suggesting that, in this huge group, there might be a subset of blastocystis organisms that do cause symptoms where treatment can make them better. But the vast majority of time, it's a commensal. It's not causing any issues. And that's actually sort of what my thought was after this treatment. We see Endolimus xana, which really does not cause symptoms. There's that very interesting case of the gentleman with advanced immunosuppression who ended up with an invasive Endolimus xana tumor, right?
Yes.
And it was very exciting. I think I was in southern India, like right as that paper came out, and that was like what we were all talking about. I was hanging out with like-minded people who want to talk about such things. And then we have that blastocystis. So my take on this was that this was a dysbiosis being triggered by fecal exposures. And when I got into the details of these social interactions, they involved sexual practices that had a fecal exposure. And that's really seemed to be the timing. When he would have a successful encounter, engage in one of these particular sexual practices that would put him at exposure for fecal material, then he would have this dysbiosis. Sometimes he would end up with a pathogen, right? A couple of times he got Giardia, which I think were true pathogens. Treated, got better. Giardia, treated, got better. But then he developed this. Treated, no impact on treatment. And that's what I think it is. That was my clinical impression in this case. And he's actually someone I continue to work with a gastroenterologist, trying to restore a proper microbial gut balance.
So the title of that, TWIP 99, you get your polar bear from your Nana.
Okay.
What was the basis for that?
So when, one of the things that we have to do in our training, and Dixon probably, is that you have to look at under the microscope and identify the different organisms. And so there's a polar body. And so that's one of my mnemonics.
That's great. That's great.
Yeah.
Well, so if you're interested in what I think, I remember.
We're very interested in what you think.
No, it's okay. You're going to hear it anyway. You have talked about it before. That was one episode. And in the Parasitic Diseases lecture series, there's a lecture just to cases where it's tough to figure out what's going on. And these were two of
That's right.
the hosts, the microbes involved. So then looking at other cases, this is often seen, right? Gastrointestinal problems. You get both of these isolated. There's nothing else unusual.
Right.
And, there's the whole flora, which it's there, so people don't make any note of it. And it often happens after sexual encounters. So I would say, yeah, these two somehow together are causing this imbalance. But maybe, many people have NANA, right? Many people. So maybe together with blastocystis, that causes it. So I thought these two organisms were the diagnosis, but it seems to me that it's not. It's more like a dysbiosis, and I think that we don't really know in the end, right?
Yeah, that's right.
Yeah, I mean, that's true. I mean, now that we've gone ahead and we've got them to start using kefir and yogurt and, different things, a probiotic approach, we're seeing some improvement. It's sort of, but I will say, and this is interesting, I don't know if this is a case we ever presented, but we had a gentleman who went to South Africa with his wife, and there was a water shortage, which, happens a lot in South Africa.
That's right.
And so there were issues with hygiene, and he came back with blastocystis, having, horrible diarrhea. He was treated, and coincident with treatment got quite better. So I do think there are certain subsets of blastocystis. I think, there's sort of genetic studies suggest that. But a lot of times, this is something clinicians have not seen before. The tendency is to treat it. You don't always necessarily need to. And like in this case, the treatment didn't seem to make a difference.
Right.
So you just told him to have kefirs and...
Yeah, we talked about how to restore his proper gut microbiome. We also talked a little bit about, he enjoys this particular sexual activity. And, as we established a pattern, it really seemed coincident. He'd get better. He'd be feeling great. Then he would go ahead and get the same exposure. And there wasn't always anything exciting in the stool, except for he would have symptoms, and they would last for a 3-month period of time. And so.
Can I ask, is when you see symptoms, so, in men who have sex with men, there are the insertive and the insertion, right? Mm-hmm. Are those the right words?
Yeah.
So does it correspond with either one?
It really was sort of an oral anal style.
All right, okay.
That's
that makes more sense.
Exposure.
That makes more sense.
Yeah,
there's not much you can do about that, Dixon.
nothing except caution them. And you're doing your job, and if they don't want to listen, that's their fault.
Yeah, sure.
The other caveat on this one is that if this person was not gay and did not acquire this through his sexual activities, these commensal organisms that are acquired through fecal contamination could be indicators of microbial life that is not parasitic, that is not, they'll be bacterial rather than protozoal. So you'll do stool cultures as well to look for things like Shigella, Salmonella, that sort of thing. So it's good to follow up on them. And a lot of times they did turn out to be the cause of their illness.
One last question. So you should see this in women then.
Yeah.
It shouldn't be restricted to men, right?
That is true.
Do you?
Yeah.
Oh, okay. Makes sense. All right.
I've got one question as well, Daniel. I did read that you could use alternative drugs as well to treat this. Did you attempt to do that at all?
Yeah, that's interesting. And I think the one emailer brought that up is because what, alternative suggestion would be that we just have a blastocystis that is resistant to the metronidazole. So blastocystis, maybe we have, and I would love, right, if we could genetically type this, determine if it's one of the ones associated with pathogenicity. But the other is, do you go ahead and use an alternative treatment?
Such as paromomazine.
But I guess then that wouldn't really kind of tally up with the coincidental appearance of the symptoms after a successful encounter.
Yep. And the fact that they tend to have this cyclic, they would resolve over time. And, and we're trying the probiotic approach, working with the gastroenterologist.
Yeah.
If he doesn't get better, maybe symptoms get worse. That would be reasonable to consider. But
Boy, it's been a while since we were all in the same room. We used to be in the same room, Christina,
true.
when we recorded TWIP. We
is
sat
true.
around a table, a round table, and I miss that. It's fun because you can see people's expressions so much better than
Yeah.
on Zoom
This is
for
all
some
true,
reason.
all
I
true.
don't,
Yeah.
you know.
Yeah, you've actually got a really nice photograph of the three of you sitting at the table on the Parasites Without Borders webpage.
Yeah.
This is
Yeah,
true.
that's nice.
Sadly, it's
yeah.
not going to happen
Yeah, I
anymore.
kind of find you can't really see so much when someone wants to speak so easily, or because you can't hear the intake of breath, maybe, and it's
Yeah,
a bit more difficult
sure.
on Zoom. So
But you know, Christina, for years we did audio only,
Right.
that's
Yeah.
true. Yes, of course.
and very hard to hear when someone wants to
Mm.
talk. So often you step on each other all the time, right? We'd interrupt each other.
I feel like we
Yes,
did
of
it
course, yeah.
backwards. Like now that we're doing the audio, I'm sleep deprived. I have permanent bags under my eyes. I was a better looking man back in the audio days.
You get older. We're all getting older, Daniel. What are you going to do? All right, we have a paper for you. This should be fun. This
Oh,
is
this is-
Dixon's choice.
Actually, I will modify that.
Okay.
My wife, Marlene Bloom, whom I share my life with, was sitting at the dinner table and reading something from the New Yorker magazine, I believe, and it mentioned this paper as a confounding principle of life's continuous exploiting of the possibilities of interactions. That's as vague as I can be. And then she read me the sort of the abstract to this, and it sounded perfectly reasonable to look into it further, and I did. And when I did, I discovered that it was much more complex than the little blurb that she read to me. But nonetheless, we're up to this. So I said, why don't we just review this paper and see where it takes us? Because you will see from our discussion that it doesn't exactly stick with the text of the paper itself. And that is the whole point of this, is to show you, in some ways, the difficulties of carrying out experiments when you take them from the laboratory and move them into the field.
So this is a paper in molecular ecology. It's called Long-Term Spatiotemporal Genetic Structure of an Accidental Parasitoid Introduction and Local Changes in Prevalence of its Associated Wolbachia Symbiont.
I'm sorry, would you please repeat that?
Yes, indeed. By Duploy, Nair, Nyman, and Van Nuys from Lund University, that's in Lund, Sweden, University of Helsinki, and,
Your old alma mater.
well, Department of Ecology and Evolutionary Biology. They left out the university, Cornell University, Ithaca, New York.
Right.
Oh, that's so funny.
There's no, they left
True.
it out.
Not only that, that was the birthplace of modern entomology.
What's his name?
No, that was the birds. That was the
Oh,
ornithology too.
the ant guy who was up
No,
in
that's E.O. Wilson, but E.O. Wilson is at Harvard. That department in Cornell
it was
became
a-
famous.
There was an insect guy at Cornell who you always talked about. Who was his name?
Yes, he was a chemical ecologist, and that's okay. My Rolodex is turning slower nowadays. I'll come up with his name. Don't worry.
Anyway, the participants in this paper, 2 out of the 3 we've actually talked about on TWIV. We've talked about parasitoid wasps
Right?
and Wolbachia, but we haven't talked about this butterfly, which is
No.
part of the.
We have not.
So, Dixon, you want to start us off here?
Well, the premise of this research venture is to try to establish a population of butterflies on an archipelago of the country of Finland that doesn't have this butterfly to begin with. They want to introduce a species and see what happens to it. So it's a little adventure in ecological invasion, basically. But in order to do that, they have to collect these butterflies from someplace. And so they go to other islands that have this butterfly. And how do they introduce it? And that's the problem number one. Problem number one is that how would you introduce a butterfly into an area that doesn't have them? And so you have choices. You can start with the eggs. You can take them off leaves that they deposit them on, and you can take the leaves over to the island, and you can allow them to hatch, and then you can allow the caterpillars to grow up, and then they will produce adults, and the adults will mate, and then you've got a population to study. That's one way. They didn't do that. Another way is to collect caterpillars from the environment and just take them because they're easy to see, and put them in a little jar or a little cage, whatever you choose, with lots of leaves to keep them happy until you get them to this island, and then open the cage and allow them to escape, and hopefully they will choose a similar plant, or maybe that same species of plant is on that island as well. Maybe they've looked into that part. And you've introduced the species this way. And the third way of doing this is to take adult butterflies, males and females, before the mating season, just newly hatched, and take those to the island and release them and start your populations that way.
So I was going to ask, couldn't you do the chrysalises? Apparently I get
Oh, I'm sorry. That's right.
Yeah, because I get criticized. Like I'm explaining to people like how to go in and out of the airborne isolation rooms with
Yeah.
COVID. And I would say, just think about like when you're going in and out of a butterfly pavilion. And people always look at me, Sienna Moran, when I ask you doctors, like, Dan, that's not a good analogy. Most people are not going in and out of butterfly pavilions.
That's right. But those who
But I
have small
think they
children
do.
do.
But I think they do. They use they transport the chrysalises,
They do.
and that's how you establish them into different
That's
butterfly.
right. That's right. That's right. So you could do that. You could collect the chrysalises and then take them, or chrysali, depending on how you want to pronounce that, and let them hatch. That's a little risky, though, because they all might not hatch, and they may have predators that would eat the chrysalises that would recognize them, like birds and things like this. Or you can begin with a laboratory colony. You could have a butterfly room, just like Daniel said, and you can allow them to reproduce and produce all their stages. And then you can start with the simplest stage, which is the egg. I would have picked the egg, I think. And take a leave's worth of eggs that they lay on the bottoms of the egg, and then let them go into the environment and start from scratch. Or a laboratory colony, start with that to make sure that you can avoid something like what happened in this paper. So what happened was that they took wild caterpillars from one place, and they introduced them to the island. And inadvertently, they also introduced some of the parasites that infect this butterfly species and may be responsible for why it's not found on that island to begin with. These are called parasitic wasps.
Parasitoid.
Parasitoid wasps.
Parasitoid.
Okay, fine.
No, that's important, right?
Okay, well, they're also called ichneumid wasps.
Ichneumid, yeah.
They're all the predatory parasite oriented. What they do is they catch a victim and then they insert their ovipositor inside the victim, and then they lay eggs inside the victim.
Right.
The
So
victim-
how did the wasps get there too? Like you think you would know, right? You got the eggs, but where are these wasps?
the wasps' eggs were inside the
Yes,
caterpillars.
right.
They were like the Trojan horse type of thing. So the caterpillars, unbeknownst to the investigators, carried with them their parasitoid wasps. And not only that, there isn't just one parasitoid wasp to deal with here. There are parasitoid wasps that parasitize other parasitized wasps. So you've got, like, they liken this thing to a nested set of Russian dolls.
You know what those are called? The second wasp? Dixon?
Go ahead, you'll tell me.
Hyperparasitoid.
Yes, Christina.
Hyperparasitoid,
Yeah.
that's right.
Yes,
Yeah.
right.
Now, what if that also had a parasitoid?
Oh, it goes
No.
on and on. It's turtles all the way down, Dixon.
That's right, that's right. Dr. Seuss was right.
Dixon, the butterfly is the Glanville fritillary butterfly.
I love the name. I love that name.
This is, so in retrospect, they should have checked the caterpillars for the parasitoid wasp eggs, right?
I think
Well, I'm not sure, because
You
if you
can't
look
do
at the
that.
maps, actually these parasitoid wasps and the hyperparasitoids,
Yeah.
they were already very common and, very,
Okay.
I think that they were essentially everywhere on that archipelago already, if you have a wee look at that map.
yes. Maybe.
And
yeah,
I
so
think
they
that was
would have.
before 1991, so that was actually a long, long time ago
Yeah,
that they
yeah.
introduced those
then it's-
butterflies
then
together with
we're-
all their parasites
Sure.
and the Wolbachia.
Would you also consider then this as a flawed site to select? Because
No,
if
you
you want
couldn't.
these to actually successfully reproduce and maintain a population, then you should pick a place that's so remote that only it can survive,
Yeah.
apart from its parasitoid wasps.
Well, it did survive. So.
Well, no, of course it did, because
was
it
okay.
felt a balance between
Yeah.
the infected and uninfected ones, because
And
the
I think the
little
parasitoid infects about 30% of the
Yes.
butterflies only.
So
So
let-
there's only ever, it's not like in the nemat. No, sorry, I'm jumping ahead there.
No, that's
I got my
okay.
Wolbachia mixed up with the wasps. But I think it's about 30% of
Yeah.
the, what are the things called? Butterflies.
Yes.
that are
butterflies.
infected.
Lepidopterans,
are
please.
infected with the parasitoid wasp. So
But I just wanted to take a diversion into this parasitoid because we have talked about them on TWIV, because when the wasp injects the eggs into the caterpillar, the eggs produce viruses that immunosuppress the caterpillar. And those viruses, they're encoded in the wasp genome, which is amazing, right? the wasp delivers them along with the eggs. So the genes are integrated into the wasp genome. In the ova, the virus particles are made. They go in with the egg, and they immunosuppress. They produce proteins that immunosuppress the caterpillar, so they don't reject the larva. Otherwise, they would reject them. Isn't that insidious?
It's amazing.
So, what's in it for the virus? It gets a new host, I guess.
Yep.
And you may say, when did this virus pop onto the scene, right? Because what were wasps doing before that? It's really interesting. It's a fascinating
Well,
situation.
it's amazing that, with science, how everything is connected, because we sort of silo sometimes, right?
We
like,
do. We
I'm a
do.
virologist, I'm an immunologist, I'm
right.
a
Or
parasitologist.
an entomologist.
But to understand a lot of these, you need a team, right? You need people with all the specialties communicating, talking.
Sure.
That's right.
So-
That's
So, Dixon, what did they find? It is a 20-year-old study, right?
It's an ongoing 20-year-old introduction, and their results were unexpected, and they explained them in ways that made it very difficult for an uninitiated molecular ecologist to actually follow closely, because they did a lot of genetic analysis. And what they were looking for was expansions and collapses of populations, and they were trying to come up with genetic explanations. And they did fabulous genetic analysis of not only the butterfly, but also its parasitoid wasp, and then the hyperparasitoid and the Wolbachia. They looked at the genetics of all four players that regulate populations.
All right. So, you just mentioned Wolbachia. So, where did that come in? Where does that come into this
I
story?
believe the Wolbachia is injected into the parasitoid wasp by the hyperparasitoid wasp, and that kills off the parasitoid wasp and limits the populations of that parasite of the caterpillar, so that the hyperparasitoid wasp is actually helping the butterfly expand their populations.
And all
That
of
was my
these-
understanding.
All of these came in together. The
All of them
Wolbachia,
came
the
in
hyper, the
together, yeah.
parasitoid, the butterfly, all
The
came in
the
together.
entire entourage of networks of life for these four forms, of course, were introduced, and inadvertently. That was the other point. This is.
I think that, sorry,
No,
Go
no,
ahead. Go.
I just
I
wanted to
think
make
the Wolbachia, they actually kind of make the parasitoid wasp more susceptible to infection with the hyper, or parasitizing with the
Yes,
hyper
yes, that's right.
parasitoid wasp.
That's correct.
I thought that was quite interesting. So I actually that came out, the Wolbachia in the parasitoid has quite a high cost, really, for those
Very much so.
little wasps.
So it means
Yeah.
that the rate of infection in the parasitoid wasp is not 100% from the hyperparasitoid wasp, because if it were, then obviously they would all be dead. And then they would have no more hosts to infect, and therefore they would become extinct, and that would leave just the butterfly alone. But that didn't happen. So there's balance between all of these pushes and pulls, evolutionary pushes and pulls, which have, in the full explanation of this paper, which is very complicated, a genetic explanation. And it's all to do with bottlenecks of population explosions and collapses due to the acquisition or the immunity that's conferred by, let's say, no, I'm just projecting into the future. Go ahead, please correct me.
Yeah, no, I mean, I guess my this is definitely a very complicated paper, and we've talked a little about it. Very challenging to look at these issues. So they really sort of took an opportunity to observe And what you see, and I don't know if this is open access. It's open access, but
I think so.
there's a figure 2 where they're actually following the butterfly populations in the different regions, and they have them color coded. So you've got your orange, your green, your yellow, your blue, your red, and they have a table below it because they're making the argument that you see an increase, a drop, an increase, a drop, and each one is sort of a selection point when you can ask the question is there a fitness cost to Wolbachia that is correlating here? And they're going to claim that there's a fitness cost to having Wolbachia. So you can sort of look at your table, ask a question of how much, what percent of the population is, do we call infected with Wolbachia? I almost feel like we need it has a Wolbachia symbiote, has an infection.
Symbiont, symbiont's
has
good.
a Wolbachia symbiote, because it is interesting because we use these terms sort of almost exclusively, but a symbiote, there might be a certain advantage to the symbiote, but there's also a fitness cost to this symbiote.
Sure.
And I think that's sort of what they're trying to suggest. I think suggest is the strongest term you'd use in this kind of a study.
That's right.
So Dixon, they were originally interested in this idea that when a population is introduced in a new area, usually you don't have a lot of them,
That's true.
and that's where the bottleneck comes from, right?
That's
There's
the
a
one. That's
genetic
the first bottleneck. That's right.
and so it's an interesting study to do to see how the population changes
Sure.
when you introduce it.
That's right.
And I think what they were surprised at is there was not a lot of change, which is what they looked at with their markers and so forth.
that's
And
right,
I think
that's
the WASP and the Wolbachia kind of explain that in
That's
part.
right.
Right?
It does. And I mean, do you mind if I drop back a little bit in history of the idea about reintroducing or introducing species into a sterile area and then following what happens? Sometimes in nature, luck favors the prepared mind, as Louis Pasteur used to say. And there was an incident where a bunch of wolves were chasing a herd of moose. This was northern Minnesota, near Duluth. And there's an isle off the coast of that state called Isle Royal. And the ice was obviously formed across the Great Lake, Lake Superior. And the moose ran across to Isle Royal to escape from the wolves, which they did not. And then the wolves caught up to the herd and started to cull moose and to obviously do what they do in nature, and that is to regulate populations. Then the following spring, when the ice melted, there was no way for the moose or the wolves to go back. They were stuck on Isle Royal, which had no moose or wolves to begin with. And that study is ongoing now. I mean, that may be 50 or 60 years old for that study. And they've been watching populations of moose and wolves go up and down, and they call it the moose, spruce, wolf triangle of when there's a lot of moose, they eat spruce, then they deplete the spruce, then there's a lot of crashing in moose populations. Then the wolf population crashes because there's no more moose. Then all of a sudden the moose come back and it's crazy stuff. This is a study that everybody gets interested in ecology for some reason because it's like an invasion of species into new areas. And there are books written about invasion of species. And so this is a follow up on that with a much more sophisticated way of analyzing the data, because in this case, they've got genetics to back them up. Genetics is complicated. They're looking at mitochondrial dependency, satellite DNA dependency. There's a lot of stuff in here that I glossed over, unfortunately. I must say that I didn't dig deep enough to try to derive more meaning from it. But the main thing I got from this in the end was that if I'm part of this group and we're sitting around a table and we're discussing the results, we could be there for a year. The results are open to interpretation. You cannot control what nature does after you've started the experiment. You're watching to see what happens after you've set the clock in motion, and you're an observer at that point. You're not manipulating the populations. You're simply beginning the experiment and then let the games begin. And it's a remarkable study from that point because it's very difficult to do these studies. Imagine going out into the field and collecting all of these life forms and then bringing them back to the lab and then grinding them up and getting their DNA and then trying to make meaning from this that has something to do with the crashes and expansions of populations. And it's the way life behaves. So I admire their courage for just trying this. I mean, it's not and it's how long has this study been going on? For 20 years.
I think, yeah.
20 years. And Notice the lead scientist is an entomologist. And
I think the original experiment was kind of abandoned, and this study just kind of took advantage of that experiment of rewilding this small island with the butterfly, and has actually then taken the opportunity to look what happened to the various populations
yes,
on this island and other islands. And actually, there's so much detail in that paper. I spent an entire day just kind of looking at it. It was just so amazing. For example, if you look at that little map, you can see 3 islands that are close together.
Yeah.
Yes,
So the one in the middle,
that's right.
Sotunga, is where they released the
That's right.
butterflies originally. And then you've got these other 2, and a kind of by genetic analysis, they could explain, in which directions they migrated just by looking at the genetic,
That's right.
at the genomes of these creatures.
Exactly.
And for example, they could explain that there was not really any migration from Sotunga to the lower island, which is called Föglö, although I'm not sure if my Finnish is up to scratch here. But there was migration to the upper island, and one explanation they had for that I thought was interesting. It's because of the prevailing winds.
Yes, yes,
Yeah.
yes.
Because obviously those parasitoids and the butterflies, they can't really cover that much distance over open water. So I thought that was really interesting. And they also looked at the kind of the Wolbachia infection rate on the different islands. And if you kind of look at the top island, where you don't have the hyperparasitoid wasp, actually on this island, the parasitoids were almost 100% infected or parasitized or symbiosed. I'm not sure what the right word is with Wolbachia. So 90% of all the parasitoid wasp contained Wolbachia, but on the other islands it was more like 30 and 25%. So huge differences, and they actually tried to explain it all. It's all hidden in my notes somewhere, so I can't easily find the explanation now. But I just thought there's so much richness of data in there.
I would agree.
I think it would take a year to discuss, even in a group meeting or in a lab meeting.
So Dixon, where's the parasitology here?
Well, I mean, the parasitology, if you're looking at an ichneumid wasp egg as the parasitic stage of that life form, and it gets inside of another animal, and it starts to eat and grow, and essentially consumes a living organism in the process. It reminds me a lot of other relationships like that. For instance, there are lots of examples of wasps that take spiders, for instance, the mud dauber wasp, and it captures a spider. It doesn't kill the spider. It paralyzes the spider. And then it puts it inside of a mud-encased nest and then lays an egg. Of course, the egg hatches and finds the spider and crawls into the spider and eats the spider, and that's its meal. So that when it hatches, it's going to look for spiders, right? But I wanted to say something more general about studying life forms on isolated islands. Think back. Who was the first person to establish this as a methodology for looking at evolution?
Was that Chuck?
It could have been. There is a strong resemblance
Yeah.
to the very original experiments of a naturalist whose father was a pastor back in England and wanted his son to experience the world so he could explain it better to his parish when he took over the practice.
Yeah.
And the Galapagos Islands jump out at you.
And
when
this
you
is
look
the
at
man
this.
with the proper shaped head.
It's the one, and
Yes.
he doesn't have much hair on the top, and he's got
Yeah.
a lot at the bottom.
Yeah, no,
And-
that's why he was selected by the captain, because the captain checked his head.
Oh, is that what it was?
Yeah, and he had the right shaped head. The original naturalist, his head did not pass muster.
I see. So Master and Commander is a film with a wonderful actor whose name I'm blocking on right now, but you will recall his name. Vincent. No?
Yeah, I know who you mean. I'll look it up. Go ahead. Don't talk.
He's an Australian actor, and he was a masterful captain of the ship, and on it was a naturalist, and they stopped at the Galapagos Islands.
Russell Crowe?
Russell Crowe, that's exactly right. And I was in love with that movie from start to finish. And to watch this naturalist stumble over these islands with his cages of animals and his dead this and his live that, his plants these, and bringing them all back to the ship. He almost missed one of the ship's exit points and they had to come back to get him. And it was a wonderful portrayal of what it was like to be a naturalist on some of these adventurous ships that shipped out, like the Beagle. This is a modern homage to the very beginnings of these studies to see how life behaves both in isolation and then put into a different situation. Because let's face it, in the world we live in, there's a lot of encroachment. And there's a lot of introduction of species into places where they never were before. Like us, for instance, when we get involved in the tropics, for instance, and you build a road from, let's say, Belém to the interior to establish a new city, like the capital of Brazil, which I'm blocking on right now, Brasilia, of course, why would I block on the name of that? So building that road exposed all of the workers to organisms from wildlife that could now take advantage of a new host as they progressed towards the center where they were going to build us. So these encroachments and invasions and creating ecotones, as they're called, differences between one ecosystem and another, the border between the two, that's where the conflict comes in. So establishing populations on islands that don't have them creates a situation where they can duke it out with who's ever there already. And if nobody is there, they own the island. They are kings. And then that's the way that comes out. But their populations are regulated by other life forms, which, oh, you think this is your island? Well, I own you because, that sort of thing. And it's so much fun to look at the depth at which nature has evolved in order to regulate itself from running away with the whole planet and just filling it up with E. coli or filling it up with a fish species, or it doesn't work that way. Yeah. It doesn't work that way.
I like that connection with the Darwin because, we've become like sort of snobs of randomized control experiments where like we only have one variable, and here it's just the fact that you can actually learn a lot, generate a lot of hypotheses, as Darwin did, just by looking, by observing, by trying to come up with ideas of why would this be this way?
Exactly.
Why would this happen?
Exactly.
Why do certain organisms have more Wolbachia, some have less? Anything about Wolbachia is going to help us understanding parasites because it's such an important organism. And here they're observing. They're not messing. Someone messed at some point. Someone introduced, and then they're looking at and saying, what can we learn over the 30 years? Right, 1991 it gets introduced. So. I think that this is, and you see the challenges. This is not easy.
This is very difficult.
Yeah.
And not only that, there are no controls in this experiment because it's not an experiment.
Yeah.
It's an adventure. And I wanted to come back to something Daniel just alluded to, too, because he said, and Wolbachia is such an important organism. Remember that with a parasite that people used to catch a lot in West Africa called Onchocerca volvulus, it's a black fly transmitted worm infection that results in a nodule in your neck or your arm or your leg. The adult worms live in the nodule, but they produce larvae that migrate under the subcutaneous tissues until they finally find their way into your eyes. When that happens, you start to go blind. They infect all the parts of the eye, and eventually the eye coats over with these cataract-like opacities, and the person goes blind. And it's called river blindness. What is the cause of river blindness? It's the worm, of course. It's the larvae, of course. And it turned out none of that was true. What was the cause of their blindness? And the cause of the blindness was the Wolbachia in the larvae. If you knock out the Wolbachia with a drug that attacks bacteria only, and you infect an animal with an equivalent infection, no blindness. The worm doesn't cause the blindness. The Wolbachia causes the worm to cause the blindness. And the mechanism for that we don't understand because maybe Wolbachia triggers a toxin that the worm can produce only with Wolbachia there and not without. And I had the privilege of meeting the person who actually discovered the Wolbachia in the larvae of Onchocerca volvulus, and he didn't know anything. He didn't know what it meant. He didn't know anything. He was an electron microscopist. He was not a worm biologist. But he discovered it, and he's eventually given credit for that. So that's a great example of this is a totally unexpected role for this bacteria to be playing in a worm. And then there's all these other things where they sterilize adult female mosquitoes and that sort of thing. So Wolbachia is probably the ruler of the world as far as we know. I mean, it's the underlying reason that everything behaves the way it does. And how does one organism get so
It's really, really
powerful.
fascinating, actually.
Yeah.
There's and actually the kind of the relationship with Wolbachia with arthropods is slightly different than the relationship of Wolbachia with nematodes. So I think in the nematodes it's always a symbiosis. So the nematodes need them for survival. For example, the Onchocerca, I think they get what do they get? I'm just trying to remember. Some essential amino acids they derive from the Wolbachia metabolism,
Okay.
and actually the Onchocerca cannot reproduce without the Wolbachia, so they are really dependent on each other. But in arthropod, I think the relationship is more parasitic. It's not probably how they can feminize the mosquitoes,
Yes.
or how they can kind of kill off the male offspring, and that's not very symbiotic, really. That's kind of quite deleterious.
That's true. That's true. By the way, the guy who discovered it,
So actually, that even though it's one and the same bacteria, and there's lots of different lineages, but the roles in these different other phyla is really quite interestingly different. So more symbiotic in one area and more parasitic in another.
Yeah, I'm thinking of
another
the
phylum,
filarial worms
so-
like Bucheria bancrofti. The females, you destroy the Wolbachia, the females lose the ability to reproduce. So.
Right. So the guy who
Yeah.
discovered that is Bill Kozak, by the way. I remember his name.
Oh.
now.
Okay.
William Kozak.
Yeah.
And do
And
you,
you know,
you probably have on your test, right? Like, what is the one West African nematode that doesn't have a Wolbachia symbiote?
I was just going. That was just going to be my
That's
next
gonna be your
question. I've
board
always
question,
wondered
right?
why Loa doesn't contain Wolbachia.
right,
It is
that's right.
interesting though. Why does it not contain any Wolbachia?
Well, maybe because it's transmitted by a different kind of vector.
Yeah,
It's
maybe.
transmitted by
Yeah,
a
but
tabanid fly, right? Not a black fly, a tabanid fly.
Yeah, a deer.
So
yeah, but then
maybe the deer
Bucheria
fly
is-
doesn't
is-
have. Maybe the deer fly doesn't have.
Maybe
any
the deer
Wolbachia.
fly doesn't have it, but I think the transmission is mostly vertical rather and only very rarely horizontal. So maybe it's just not happened. Maybe once it's established, it will kick off, and that hasn't happened in Loa Loa. But really interesting, actually.
It's interesting also that you can introduce Wolbachia into a population of insects that don't have it, and they will now have it. And then you can use that as a control measure for this
Yeah,
male
I think that's
thing
what
that
they're
you were
doing
talking
with
about.
dengue, isn't it?
Yeah,
in
absolutely.
Brazil?
Absolutely.
Yeah.
Releasing
yeah.
the Wolbachia infected.
Yeah.
That's right.
mosquitoes.
That's
Yeah.
right. That's right.
Yeah.
So Daniel, you can treat some of these parasitic infections with antibiotics to get rid of the Wolbachia, right?
Yeah, so doxycycline, interesting enough.
doxycycline.
Yeah, so you can actually just go after the Wolbachia. And I had a case of mansonolosis, right? Not very common, right? And we had some challenges treating it, and we ended up using doxycycline. So going after
Interesting.
going after the Wolbachia.
Cool. Dixon, why is it a parasitoid, not a parasitic
You
wasp?
know, I would well.
Do you know, Christina, why the name?
No, I noted exactly that very same question. Brian. I said, why is it called a parasitoid and not a parasite?
Yeah.
Well, I think I actually don't know. I don't know.
Well, the
It's
the
more of
larva
a
kills
predator,
the caterpillar.
but it's
right?
more of a predator than it is a parasite.
Yeah.
Right. So they usually classify predators as animals that are larger than their prey. Right, like a lion that goes after, let's say, a gazelle or something. But in this case, it's a tiny little
Okay.
larva that will
Okay.
eat. And you've got the answer.
I got the answer. So
These-
parasitoidism,
Yeah.
they feed on a living host, which they kill,
Yes.
typically before it can produce offspring, whereas conventional parasites usually do not kill their hosts.
That's a bad
And
definition.
predators kill their prey immediately. So
Yeah.
that's the three. You don't
Well, you
agree with
know-
that? No?
Well, can't you name some parasites that kill their prey? That kill their hosts?
Yeah, of course.
Lots of them.
Yeah, we've talked about them all the time.
Lots
here,
of them. So that's probably
Interesting.
not a good way to differentiate.
But I was also thinking the predators. Like I'm thinking the wolves going after
Oh,
the
yeah. Oh, yeah.
the larger
Moose.
animals,
Yeah,
right?
sure. They kill 'em right away,
So
yeah.
they're parasites.
But maybe a conventional parasite is transmitted before it is killed.
Maybe.
Interesting.
So some predators can't kill by themselves. They need to hunt in packs. And
Yep.
the lions are definitely like that, and so are the wolves.
Yeah.
And coyotes. Coyotes don't hunt in packs. Coyotes eat rodents. So they're kind of along the side over here. It's a false definition, basically. I think it should all be called parasitism of some sort. That's what I think. And I'm also wondering whether or not a parasitoid wasp's egg doesn't kill its parasite. It doesn't kill its host, but rather eats what it needs and then leaves, and then the rest is okay. The organism that's been half-eaten manages to survive somehow.
You mean the half caterpillar
Are there,
wiggles away happily ever
it's
after?
either a cata or a pillar, but it's never the caterpillar. Because there are always gradations of these relationships that you can find if you look
sure.
hard enough for them. So, this is such a fascinating discussion to have on
Well,
200.
it's very interesting, and I was worried because when I read this paper, I have to admit I had the biggest problem with the names of the Finnish islands. I couldn't remember them.
Yeah, exactly right. No reason you should.
And then the overall picture I get, but I still find it confusing. I'm not sure what the mechanism is, and that's what I'm about. But
Sure.
you don't always get a mechanism, right?
But there's a wonderful general ecological niche to put this kind of study into, and that's called island ecology.
Oh yes, absolutely.
So that's another Darwinism, by the way.
Yes.
Darwin almost proved that creationism doesn't exist based on his study of volcanic islands that were in various stages of population growth.
In fact, David Quammen's book, The Tangled Tree, is all about island ecology, right?
Sure,
All
sure.
of these naturalists going from island, from back then to the present,
Sure.
and still doing it today.
Yes.
Because it can be isolated islands are fascinating, right? Because they're isolated.
Yep.
And in that book, he told the story of Krakatoa, right? Which an island in the
exactly right.
Pacific that blew up. It was
Exactly
a
right.
complete nothing left but a few specks, and then it came back
Yes.
through volcanic activity, and then years later, it was completely full of life.
Exactly.
Plants, seeds flew in,
Exactly.
and somehow birds got there, and other animals. Animals can float on bits of wood long distances. It's amazing.
Absolutely right.
It's
Absolutely.
absolutely amazing.
Like the saltwater crocodile can swim 500 miles. That's incredible. And coconuts can float forever, basically.
Yeah.
And turtles can float long distances
sure.
on their backs.
Sure,
Apparently, they're happy. Daniel, there's Plum Island off the end of Long Island.
Oh, yes.
There's a high containment laboratory there, and between the island and the mainland, there's Plum Gut, which is very rough.
Right.
And they say deer swim across it.
Exactly.
There's actually a guy that's rotates who actually watches and they shoot the deer because you don't want the. So I don't know if people know this, but Plum Island is one of our biocontainment sites. There's lots of stuff there that we don't want on the mainland, and the deer potentially. It's not a far swim. I've sailed through there, and it's not a far swim. The deer can swim back and forth, but you don't want the deer going over.
Yes.
Getting something swimming back.
And in fact, it's a laboratory for animal pathogens, not humans, but like foot and mouth disease
That's right.
and
That's right. Rindapest and stuff.
avian influenza. So they don't want the deer getting them and bringing them back to the mainland.
Yeah.
Exactly.
All right.
This rings a bell. Did you discuss that on TWIF before?
I probably did. I probably repeat myself all over,
No. It just kind of rang a bell.
Well, I used to go out there as a grad student. I went out a few times to do some research on avian influenza. And I remember on one trip, the guy said, this island is here because this is a rough patch of water, but even deer, you can see deer just their head above the water swimming out.
That's right.
And so I didn't know they shot them, but
Yeah, it's I'll tell a quick story and then we'll. But so I grew up sailing, and I'm about 19, 20 years old, and I have not a big boat. It's about a 26-foot boat, and I'm visiting my mother, and the storm moves in, and I'm supposed to sail from South Long Island through this stretch up to Connecticut, and it's such a bad storm the ferry isn't even running. And I say to my mom, I'm not sure about the weather today. And she says, Daniel, you're an excellent sailor. I'm sure you'll be fine. And so we try to sail through the gut there, the race, at this, when the tide is like going against us, and we're barely moving. The Coast Guard comes out, sits off our stern, watches us, waiting for us to sink, I think. Waves breaking over. We finally eased our way through and then made it back into the sound.
cool. Plum Gut. It's a great name.
So you got to wait till the tide is slack, and then you can swim across.
So, Daniel, we're at 200 TWIPs. I guess you've run out of cases, right?
This was tough because I had I felt like what is that, like the first day of school or something, where you want to make sure you pick a good one.
Yeah.
And so I do have a good one. Dixon, you can't look, you can't cheat.
Yes, I can.
So I was, going over in my head different ones, but this seemed like a fun one. So, and I'm hoping that this one is a little bit easier, that people will feel confident writing in. So, this is the story of a gentleman in his 30s. And this man does have a several-year history of HIV/AIDS, so he does have low CD4 count. And he presents with new onset lower extremity weakness, headache, and fever. Ends up in the hospital. We get a little more history. He reports that he lives with his wife and her cat. He reports that he's supposed to be taking some medications, but does not always remember to take them. Go a little more into depth here, past medical history. So the HIV was diagnosed a few years prior to admission. He has a CD4 count that is less than 100. He does have a detectable viral load. Doesn't report any surgeries, no allergies. His family history is non-contributory. As far as medications, again, he's not very good at taking these, but he reports that he was told to take a medicine every day, one 3 times a week, and one weekly. He's currently out of work. As mentioned, he lives with his wife and her cat. As far as substance use, he reports that because of his religion, he does not drink alcohol and he only eats very well-cooked meat. As far as other exposures, only local travel, but he was born and raised in Puerto Rico, in the big city. He has had multiple sexual exposures with multiple female partners over the years. On physical exam, he's following commands. He's thin. He's got a white coating, a little bit on the sides of the tongue and the buccal mucosa. Lungs are clear. Heart exam's unremarkable. Abdominal exam soft, non-tender, normal bowel sounds. Neuro exam, he does have weakness in the lower extremities, and there is some asymmetry to the exam. His skin is unremarkable. He has a number of labs done, but I'm gonna share with you the radiology. We have a CT result.
I can see this.
And we have an MRI result. The CT shows multiple ring-enhancing lesions with surrounding edema. He has an MRI which is also showing multiple enhancing lesions. Okay. I'm closing this so Dixon can't peek over and see this.
Just looking at that gave it away to you?
I didn't look at it,
Okay.
but it gave it away for me.
So
I don't even need to see it, I think.
No, I don't either.
you know what all
You
the
sort of
words
have it
mean, huh?
pictured in your mind, like which.
I think I can just see it. This is a very unwell man.
Yeah.
Indeed.
So this man is HIV positive, but he has had multiple female partners. So do we understand?
We suspect that's where he acquired it, and some of those encounters involved monetary transactions, if that's helpful.
Okay, got it.
Yeah. And
Indeed.
yeah.
Yeah.
And so what I'm going to ask of our listeners, a lot of people, because this is This Week in Parasitism, probably have a sense of what it might be, but let's try to give the whole differential, right? Because when we see a gentleman like this, it isn't necessarily that one parasitic thing. It might be something else as well. So we want to be thinking about those. Does the coming from Puerto Rico, does that matter? And then how are we going to establish the diagnosis? Are we good to go? Is there any blood work? Are we going to do any therapeutic trials? Are we going to do biopsies of his brain? What are we going to do at this point?
How old was he when he came from Puerto Rico?
So he came right when he was about 20. So he had been in the States for over a decade.
Gosh, if you Google ring enhancing lesions. The differential is like 20 different things.
So our emailers should have a lot to think about.
That's
Go
right.
for it. All right, this is one case, Daniel, where I can't ask if they're HIV positive because you already told
He
us.
is.
All right, very good.
Yes.
Dixon, I forgot to ask you if you had a hero.
It's okay. Yes, I do. Yes, I do. So this is a current person who I have deep regard for and have had the pleasure of working with in the past while he was a professor at Tufts University's medical school in Boston. And his name is Gerald Kirsch, Keusch. Jerry Kirsch, his educational history involves a undergraduate degree at Columbia University, Columbia College in those days, and then an MD degree from Harvard University, and then came back to Columbia University as a postdoc, or as a visiting research physician to work in the laboratory of Elvin Cabot, who you, of course, you knew very well as part of our microbiology department, and then got his first appointment as a faculty member at Tufts University. And while he was there, because we had met while I was at Columbia also, and we became good friends, he started to invite me to help him teach his parasitic disease courses. And I can't tell you how much fun we had. At one point, he asked me to bring up some infected mice with trichinella larvae and adults. And I did. And we got the adult worms out. And as some of you may know, the adult female worm gives birth to live larvae. And we had a microscope set up to witness the birth of the live larvae. And Jerry,
only a parasite aficionado, right?
yeah, well, we had the whole class of medical students in this big lab, and Jerry and I were looking through the microscope at these larvae one at a time being born.
Wow.
And as they were being born, I would announce, "It's a boy." And then he would hand somebody a cigar. And then he says, "Oh, it's a girl." And then he would hand somebody else a cigar. And he had a whole stack of cigars to hand out as the birth of this worm occurred over time. It excited all of the students. They all wanted to come and take a look at this. Every class that we taught during that week of intensified parasitic disease presentations was uniformly greeted with enthusiasm and attention. No one cut class because they knew that not only would they learn things, they would be entertained at the same time. He then advanced his career by becoming the head of the Fogarty Center in Washington, D.C., which proved problematic because of some politics involved in funding. And the Fogarty Center was supposed to foster international cooperation between less developed countries and the United States with regards to infectious diseases. His research interest was, interestingly enough, not parasitic, but microbiological. He investigated Shigella, Shigella flexneri, as I recall, and the toxins involved and how they affect the gut tract and that sort of thing. But always passionate, always attentive. Then later on became I believe, the research provost for Boston University. Mm-hmm. And at Boston University, he was solely responsible for getting the P4 facility established there. So, which had a difficulty in opening, and unfortunately, I was unable to make the TWIV visit that resulted in some remarkable photographs of the TWIV team dressed up in a P4 facility protection.
That was cool.
I missed that, and I regret for the rest of my life that I had never participated. But Jerry and I have remained close. I used to stay with him when I taught with him, and he is such a gentleman and such a scholar and such a wonderful person and a caring and sharing person. His children are all very, very wonderfully educated and very alert, and his wife is a pediatrician, and she also is among those wonderful people that you really want to know and stay attached to. So I don't worship him as a hero, but I know he is a hero to many other people. I don't even worship him. I value the friendship that we've shared over these many, many years.
Yeah, I met him at some opening ceremony for the needle. The P4 BSO4 facility. Yeah, very nice guy.
So we would tell jokes every now and then. I must tell you the worst joke we told, and we loved puns and plays on words. And we at one point, I asked Jerry whether he knew that there was this new designer birth control pill that came out. And he said, no, I actually hadn't heard about that. What's it called? And I said, it's called Calvin Decline. Okay, fine. We told jokes along those lines, and we just had a lot of fun.
All right. Thank you, Dixon.
You're welcome.
That's it. That's TWIP number 200. You can find the show notes at microbe.tv/TWIP. You can send your guesses for the clinical case and any questions or comments to TWIP at microbe.tv. And if you really like what we do, consider supporting us, microbe.tv/contribute. And this month, this next 2 months, Parasites Without Borders fundraiser will go to support microbe.tv, right, Daniel?
That's the whole goal. We got to keep paying the rent here on the incubator.
Exactly right. Exactly right.
Yeah, we got to get this chip on the wall fixed. You know?
You have to put the light on in the incubator.
So you can go to parasiteswithoutborders.com and you make a donation and they will match it.
And we got our first donation. We had a gentleman come by and actually hand deliver the first check of the fundraiser. So this is exciting.
This is a destination. If you want to come visit, just let us know.
Bring a check. That's great.
You have to be vaccinated, though. And
yes,
this is true.
You have to have a mask.
This is true.
Dixon Despommier is at trichinella.org. TheLivingRiver.com. Thank you, Dixon.
Oh, it was an extreme pleasure.
200 episodes you and I have done
That's
together.
That's right.
We've been through the whole thing, Dixon.
We have. And 200 more to come.
200 more to come. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Always a pleasure.
Christina Naula is at the University of Glasgow. Thank you, Christina.
Thank you for having me. I need to get myself a website, maybe.
Yeah, you should do that. Everybody else seems to have one, right?
I know. I do have a Twitter handle, but that's about as far as
What is that? You want to tell us what that is?
I think it is at Christina Naula 1, all in one word.
At Christina Naula 1. All right.
Yep.
Now you're going to get billions of followers.
I've got about 3 or 4.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWIP and Ronald Jenkins for the music. This episode, no, that's not what I do at this point. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWIP
is
parasitic.
Sick.
Oh, we were in sync at least.